Welcome.
The Pathology Innovation Collaborative Community is a regulatory science initiative that aims to facilitate innovations in pathology, as well as advance safety and effectiveness evaluation. The PI Collaborative Community aims to harmonize approaches to speed delivery to patients through collaboration in the pre-competitive space. The PI-CC is open to all stakeholders, public or private, including but not limited to academia, industry, healthcare providers, patients, and advocacy groups. Meetings of this kind are an example of lawful activity under federal antitrust law, because the focus of the meeting is on government action or policy, including the industry's responses or positions taken with respect thereto. However, antitrust monitoring is needed for companies' own protection, since they are direct competitors meeting together. These meetings need to stay within protected subject matters and need to be monitored so that they do not stray off into inappropriate areas, such as pricing and price terms, sales and service territories for particular products, customers and customer territories, each company's individual decisions regarding selection of suppliers or customers, marketing plans and especially future marketing plans or new product offerings, other proprietary or competitively sensitive information. Today's meeting will follow the written agenda, and there will be no discussion of pricing or other prohibited topics. If the discussion veers into those topics, we will terminate the discussion. If you have any questions or concerns about the propriety of a discussion, please raise it immediately, and we will take a break to determine how best to proceed. The Medical Device Innovation Consortium acts as the formal convener, and we will follow their conflict of interest policy. For additional information, we defer to our Code of Conduct and our website, www.pathologyinnovationcc.org. We look forward to moving the field forward.
Good afternoon. It's May 25th, roughly 3 p.m. Eastern Time, and I'm very happy to see everyone. Let me just start sharing my screen, and I stay here so I can see the chat. Does that work?
Yep.
Perfect. So we have a very packed agenda. As you probably know, there's been a lot of stuff going on, and first is, of course, a quick review of the Pathology Informatics Summit in May, earlier this month in Pittsburgh. You see, aside from all the science and the discussion and content, meeting friends in person was definitely great. And a big shout out to everyone who made it. I don't know if someone wants to comment. Kate, Brandon.
You see all three API presidents, past, current, and future, in the picture there.
Let me just check. That's right. 1, 2, 3. Yes. I don't know. Joe mentioned he will be a little bit late, but no, was definitely great. Okay. Moving on. Before I turn it over to Xavier Palazzi, who is on the call, and thank you for staying up, or I think there's a lot of Europeans on the group, but let me just quickly explain this critical path innovation meeting, and I think Ulla will share some of these documentation, is a pathway, a program actually, with the FDA to propose some cutting edge things. Novelties, anything from regulatory science project all the way to providing suggestions for draft new guidances or regulation. And Xavier and team have a very interesting proposition, namely to implement whole slide imaging in the regulated non-clinical development of pharmaceuticals and chemicals. And there are very good reasons. And without further ado, I turn it over to you, Xavier. If you want to briefly outline this, that would be great.
Yeah, so thank you for the opportunity to present. So I'm going to share a few slides, if I can. I think that should be coming up Yeah, are you able
Yes.
to see a presentation? Yeah, all right,
Yes.
very good. So I'm talking on behalf of a European consortium that's called Big Picture. This is an academy/industry consortium that started in February last year. The purpose of the consortium is to build a large repository of slides, clinical but also non-clinical slides. The consortium is subdivided into several work packages. One of them is about regulatory and interactions with regulators. The major component is on the clinical side, and in particular the GDPR regulation. But there's another component which is non-clinical. And so I'm a veterinary pathologist. I'm working at Pfizer. I'm in the US, actually on the East Coast as well. And I'm co-leading this work package. So as a component of this work package, we are to interact with the regulatory authorities, both in the US and in the EU. And this is in this context that I'm presenting this CPIM meeting. So actually, we're going to meet with the FDA on June the 15th in the early afternoon. And the reason for that is that a fair amount of pharma companies have transitioned or are in the process of transitioning towards digital pathology in the non-clinical regulated good laboratory practices space. It comes with a certain number of uncertainties from a regulatory perspectives. As you will see, there are some unknowns, and this is exactly what we want to clarify through interacting with the FDA. So the ultimate goal is to speed this transition to digital pathology and help us benefit from some of the advantage of the technology in the regulated space. And we see that as a first step, because the second one we have in mind is to then bring some AI tools in that regulatory space. But the first thing is to be able to read our studies first with digital slides in the GLP space. So the consortium started by evaluating what was available in terms of background, existing regulations or lack of regulations applicable to toxicologic pathology. And so a certain number of salient features were observed. And most of the literature, besides the official guidances, is coming from professional societies. So there is a clear difference between the approach of clinical versus non-clinical pathology. We are not going to be able to take the same pathway. We're not going to be able, for example, to qualify scanners as medical devices. This is not the intent. We are going to come with different options and proposals. Even there are semantic issues. There is no clear status of whole slide images in the GLP world. There's a clear status for what a specimen is, what the raw data is. So we need to define together and get an agreement on what status we can put behind whole slide images, because there are significant consequences of it. The context of use is important. So we use digital pathology in many different contexts. So it can be just for informal consultations, but it can also be when we are reading studies and we start to use the slides in a way that's going to be essential to be able to reconstruct a study. And in that case, it falls really under the GLP umbrella. And there are some specificities that we want to discuss with the authorities. Also, we use a process called peer review, which is a process by which a peer is reviewing a subset of slides, and there are different types of peer reviews. And again, without going into the details, we need to get a good understanding of these different cases to really match the validation of what we're doing, and also the recording, the archiving of what we're doing with regulatory expectations. We want to talk about the validation, and we want to talk last but not least about storage and archiving. That's a big component because there are technical consequences. So this is kind of the meat of what we want to discuss with the authorities. It's not an exhaustive list of the questions that we have, but it gives you a bit of an idea of where we want to go, because we're coming with some proposals, of course. and we'll see. We're going to pressure test these proposals and see what we can get from the discussion. So as I mentioned before, for us, the approach of scanner qualification is going to be different. We are pushing towards working with a fit for purpose kind of a validation that takes into account what we do. And without going into the details, toxicologic pathology is very different from clinical pathology through the standardization of specimens, through the comparison between controls and dosed animals, and so on. In terms of semantic, there's some sort of a consensus at the moment that whole slide images fall under the notion of a faithful replica. It's not a true copy of the specimens. It's not raw data, because for us, raw data is the signed pathology report. So we think that's probably the best way to characterize and qualify whole slide images. Another important consideration is how we manage the different image formats from what we get from the scanner, the format that might be obtained for slide viewing purposes, and the format that might be used for archiving purposes. So the approach here is to say, as long as we can demonstrate through a validation that these different formats are still a faithful representation of the original slide, they are okay. They are good for us to be considered as the tool we're going to use to derive diagnoses from and then reconstruct studies from. So it has additional implications in the sense that if you can demonstrate that from an end-to-end perspective, do you really need, or do you need at all to archive all the intermediate steps, or is that good enough that you're going to archive the final, for example, a DICOM format, and not necessarily the 2 or 3 previous steps? So that's the kind of discussions that we want to have. Also more details in what we want to put in the validation plan, what kind of documentation we want to have for the different cases of peer reviews that I mentioned earlier on. And the last point is also to understand, because we know there are differences across jurisdictions in terms of inspection for archives, because we know for specimens, there's an expectation that archives can be amenable to physical inspection. So is that the same for whole slide images? Do we need to have the servers on site for an inspector to come, open the door, check safety measures and so on? Or is there more leeway, and does that let people think about using more cloud-based systems even for the archiving component? So that's basically the topics that we want to address. And so I'm informing this group because we are planning to be inclusive, and it's trying to expand the attendance. So first, we want to see if we can bring some of the big picture partners representing the FPA, so it's the European Federation of Pharmaceutical Industry and Associations, and also a number of PIC members if there is some interest in the group. So that would be restricted in terms of numbers, and that would be under a listen-only mode. But the idea is to to have other people benefit from these interactions. And so the plan is for us to submit our slides the 1st of June, and then use that opportunity to bring that to the FDA meeting manager and see if and how we can also technically include an extended membership to the meeting. So basically, this is what I wanted to share with you. And if you have questions, more than happy to try and answer.
This is great. Xavier, if you could share those slides with us, then we can post that also on the website, and anyone who's interested in this, of course, comment. Brandon, you had a question?
I first want to say, thank you, because I reached out to Big Picture. I know them and asked if they'd be willing to expand the attendee list because I'm sure there's many people that would like to at least hear the conversation, because normally these meetings, are very one-to-one interactions. And so to already be saying we want to make a one-to-many interaction is a wonderful thing. And so I want to applaud you for that. And hopefully logistically or operationally, we can figure that out. I had another comment, but I'll hold it for a second.
So yeah, we're gonna try and see if we can record as well, and see if we can have something like a white paper as an outcome of the meeting. So, but of course, this is pending, you know, the agreement from the FDA partners.
Yeah. The other thing I wanted to make sure, Xavier, you know about, and I think I've shared it with your team, and I shared it with ICC just now in the chat, is a very basic guidance document about toxicology and digital pathology. So make sure that some of those questions that we've already answered in that document, you can check those off, because some of them were very close to some of the things you just mentioned.
Yeah, this is extremely timely, and all the companies and professional societies are commenting on the draft guidance at the moment. So there are, as you mentioned, there are overlaps or things that are very, very timely to have this kind of discussion.
Yeah, so the draft guidance is from April, so the commenting is still open. So even after the CPIM meeting, right, which served a slightly different purpose from a regulatory perspective, condensing this with several of the companies and or other interested parties on this call may be helpful. So we could even consolidate what PICC representatives are thinking into a separate statement that you can take or leave out of whatever you want to contribute. But that's kind of the idea of PICC, to provide broad stakeholder with that opportunity to provide input. And it's great if you could allow a few representatives to join in, whoever is interested. I see there are some comments that suggest that we will have some interest.
Yeah, we'll do our best.
Other questions? By the way, it's also great that Big Picture and PICC work together. And then, the second part, which I think Brandon mentioned, the CPIM pathway is, of course, open for other projects of similar sort. And I'm personally quite interested in, also supporting the non-clinical branch, which, we haven't had a lot, but this is clearly advancing or, moving innovative ideas forward. So I think this could be a great blueprint for what may eventually happen on the clinical side, but that's up for future discussion. Other questions? All right, then we continue, and thanks again, Xavier, for sharing, and it would be great to include the PowerPoint on our website update, so for those who couldn't make the call, but we'll check the project to see this.
Yeah, okay, I'm going to drop off the meeting right now and then send the slides to Ulla directly.
Thank you very much. But feel free to join, but if you have more time, but I know you're very busy, so
yeah, and
but you're welcome to stay as well.
Thank you. I don't want to interfere. Thank you very much.
Thank you. Right, let's move on. The next item is a planned meeting jointly organized with our one of our first patient representatives, Friends of Cancer Research. This is a meeting planned for Wednesday, July 13th, where Friends is, of course, a patient advocacy group, and one of their dedicated aims is to accelerate policy change and, supporting groundbreaking, mostly regulatory science. They have developed a very interesting approach to collaborate, and as a collaborative community, this will be the right place to, we've spoken to them several times, to share how they tackle things collaboratively. So if there are certain problems that are really challenging, let's pick, for example, financial sustainability of digital path. This is the topic to really learn from them, from their prior approaches, and see what we can learn from this. So I'm very thankful that Mark Stewart, with support of Jeff Allen and Brittany, will be hosting this. I think we already received a slide deck, which I think speaks of their absolute perfect organization. So if you want to join that webinar, you can basically see the slide deck already up front and come with well prepared with specific questions. So I think that would be great to share that as well. If not, I think we have the project site already up, thanks to Ulla. Questions about this? So this will be not necessarily about digital pathology, but about the approach that Friends has propagated over the years, and they share some of their success stories and experience in how to accelerate change. There are no questions, we can move forward. I think I saw Ali Bashashati on here. He's joining us from British Columbia. Are you there, Ali?
Yes, yes. Hi.
Wonderful. Ali, hey, good seeing you.
Yeah,
Do you want to tell us a little bit about your idea of the OV Scout challenge?
Oh yeah, so first of all, thanks for the opportunity. And so what we have in mind is sort of like we're starting this grand AI challenge on ovarian cancer histotype classification that we've been working actually with Kaggle on this, and then they're sort of willing to provide their platform to do this. And the idea is to there are 5 major histotypes of ovarian cancers that we want to build AI models to differentiate those, and also try to build models that I think as part of Panda that wasn't actually addressed was that, you know, identifying rare subtypes of cancers, like meaning that if you get an ovarian case, it might be one of the 5 major histotypes, but it might be some of the rare subtypes. So we are trying to address that part of the problem as well, so that, you know, it becomes, I think, more clinically applicable as part of this challenge. So what we are looking for is sort of like to incorporate, I think, regulatory aspects and get advice on those aspects to make sure that it is actually done properly. That also adds sort of value to the, you know, for the community, rather than just so in other words, it should be just beyond ovarian cancer, something that we can actually learn and apply to other cancers.
Thank you, Ali. So if I'm not mistaken, there are several people who will probably be very interested in working on this from machine learning, but also from a data quality and or, you know, data creation perspective. And there are certain pathways that, you know, from a ground truthing perspective all the way to MDDT that this may actually serve quite well. Questions for Ali or team? I think Ulla has already created sort of a project site. And if you have questions about the team and or, you know, the contact partners, that's already listed on the website. So please feel free to reach out to Ali or via Ulla.
I have a question.
Mm-hmm.
So
Kingsley, hey.
yes.
Good hearing you, seeing you.
Thank you. So is OVScout a group that's working with Kaggle, or it's the name of the challenge?
No, it's just the name of it's a tentative name. So that we've come up is just the name of the project. But this is what we have been doing, is that we're working with the ADA consortium, which represents about more than 65 ovarian cancer research teams, you know, internationally. And so as part of this, sort of we have access to data through ADA as well, and also access to other centers. So obviously, again, looking for more collaboration and more input on various aspects, you know, from this team and PyCC.
Thank you.
You're welcome.
Brandon.
You know, I just want to make sure, Ali, that you stay connected to the how to use challenges in a regulatory application conversations project. So please check out that space on the Pathology Innovation website, and just keep your radar open for when those conversations happening, because I'm a believer that this kind of environment of a challenge, where if you're going through all that effort to create all that data and you're going to hide some of it for testing, go the extra mile to, give us to save the information about the patient's population and other things like that, and the imaging information, so that ultimately it could be used in a regulatory submission, the validation test results. And I really think that is a tool that could really serve the regulatory and clinical space.
Yeah, absolutely. Thanks for the comment, Brendan. And yes, you're actually following. I mean, we've been sort of putting together a document based on like Page AI's submission as well. So that takes some of those aspects that you mentioned into account. But again, happy to work. And then that's exactly what we really want to do, to have the regulatory aspects in mind. And otherwise, the project can go live, but I feel like, we can do much, much more than this, and then just beyond ovarian cancer as well, something that is, can be used for other projects.
Yep, maybe instead of just have your radar open, if you don't hear anything, it's time to poke the beast, right? And try and get another conversation, especially if you have questions or want to engage. I would make myself available to, especially for one-off meetings at least, to answer questions and try and get to that endpoint.
Yeah, fantastic. Yeah, more than happy to do so.
Other questions? I know we're coordinating a follow-up meeting with Roberto Salgado and Francesco Ciampi. Timing is a little bit tricky because we have to, be respectful also of the European time, and everyone's so busy. So that will definitely be a follow-up. And then maybe I skip one slide real quick since you mentioned it. We have a planned event for a page decision summary event that's jointly organized between DPA and PICC. Page will contribute 2 speakers, and we just found out that through Michelle Tarver that we will get Dr. Peter Yang from the FDA as a representative from the Office of Policy and Regulation that will provide the necessary regulatory framework of a decision summary and basically how to read it. And one of the key elements there is that, of course, as more of these tools come on board, the Page decision summary will be one example that we will dive in deeper, and Page has promised that they will talk a little bit about what that looked like. Of course, that's up to them what they can or will share, but that will definitely be an event that should be of broad interest, and we will blast this out probably through all channels, because this is probably a lesson that will send a message on what to learn from existing decisions. And that probably goes beyond AI tools in itself, but just learning how to read a decision summary is probably one of the key skills, and what it can be used for. Hearing that from the experts from the FDA and from the regulatory leads from Page is probably going to be a great event. So please take note of this time. And we rescheduled this. Some other messages were possibly out there in the afternoon, but we wanted to make sure that our friends in Europe can also participate. So that will be 10 a.m., so somewhat of an unusual date, because usually PICC is in the afternoon. But just please make a note, because that should be a very good event.
Joe, is that 10 a.m. Eastern?
Eastern, yes, sorry.
Okay.
We literally got the confirmation from the FDA 2 minutes before this meeting. So
I see it too.
yes. Now we tried to get, one of the experts that's kind of like, I don't know, in general terms, people would probably call it document control or document design control. And, having someone like that as providing the relevant basis would be awesome. And so I think that this will be a very useful recording that we will obviously share also through the DPA channel. So we're also very happy that the DPA joins in organizing this. Moving on, next project that is under full steam ahead is the SEER Database project. Currently, there are 4 meetings scheduled kind of as 45-minute workgroup meetings. Briefly, the SEER database is a relatively representative database for tracking everything related to cancer and epidemiology related to cancer in the US. It's not all states, but a representative cross-section. And in the 2.0, which implies that we will make the data that we curate publicly available, plus very likely interactive if we can get everything together. The other part with 2.0 is also that we want to implement the future state, not just how cancer is diagnosed, but also some of the emerging technologies like whole slide imaging and cell-free. Samantha? I saw you.
Yeah, thanks, Joe. Sorry, I just have to find the unmute. I have a question. How does this, or does this interact with the current NCI SEER cancer path chart initiative? Because that's something that we are heavily involved in at the CAP, and I know at CDC, NPCR is also involved, and AJCC and American College of Surgeons, et cetera, et cetera. So I don't know enough about this yet, but I'm wondering how that potentially overlaps, because it sounds like there's some similar goals.
Yeah, so the goal of this particular project is basically, or it was started as a single percentage, which is, at some point someone asked, well, how many cancers are actually diagnosed using the microscope? And we discussed several things. There's NACER, there's SEER, there's the state registries in certain states, then there's the American College of Surgery that tracks cancer surgeries, as well as, correct?
Yeah.
And so basically this is a project where we asked for input in how to do this better. And through this multi-stakeholder community, a lot of people contributed in terms of saying, well, cell-free and whole slide imaging should be accounted for. Now I know that the NCI and some of the initiatives that CAP is leading is also discussing on whether we can verify cancer diagnoses, right?
Yeah.
Like, is there, for example, the option to join with, let's say, the data commons section to have actual representative images or even whole slide images to enable the full power of this? We would probably abstain from that. We pulled the data to the current state, which is going to 2019, and there are, to my knowledge, no image data available whatsoever.
Wow.
So this would be a first point to say, is it 80% or is it 94%? And I'm literally just talking about the SEER database, not any of the other initiatives.
Okay, that's super helpful. Thank you.
Yeah.
And if you need any like cross points or touch points, you probably already have them all, but just let us know for where these might potentially cross over. Yeah, it's really, really amazing.
It would be great to get collaboration and input from CAP. Brandon, I see your hand.
There is a pilot study that we heard about at Pathology Informatics. That is a pilot study for pediatric cancer. And they have like 8 sites that are very diverse. We're talking Indian reservation sites, rural, city, academic centers, places that don't even know anything about full slide imaging that are, and so data is actually being collected right now in the pilot study. And it would be so amazing to think that the goal is all cancer patients would have some representative slide included in the registry. Huge.
Yeah, this project started with one summer intern that transitioned over to another summer intern that now worked for basically 1.5 years on this, because the data, Samantha and others who worked with SEER know it's very tricky to parse the data out. The diagnostic modality is not really accounting for everything, so we may eventually propose that that should possibly be updated. And there's one other overlap with, you know, some of the cell-free projects that are being proposed. So, but it would literally be amazing to get additional input. But this was mostly retrospective, so not changing anything, but looking at all the data that have been generated over the last decade in the SEER database. Okay, other questions? So this is open to the public, so this is not just for certain folks. So if you have time and want to join, we're currently looking at the data and really just discussing how to best coordinate. That would be great. Perfect. All right, moving on. The next topic is through, again, Michelle Tarver and several of the people at CDRH and the collaborative communities, we were introduced to a group that is planning a sepsis collaborative community. And Ulla, I don't know if you have that link. This group has been started. They're called Sepsis Innovation. And this is one of those, I don't know if they're already FDA or if they have achieved FDA participation status, but they're planning to do so. And this will be something that we discussed before. This will be a start of having disease focus in collaborative communities. So the idea being the FDA has specific questions about a certain disease, that there's a collaborative community where they can get additional input. That's of course something relatively new, and sepsis, of course, we discussed this before, is one of the first topics. We're planning a collaborative meeting between either PICC organizing with the soon-to-be collaborative or both collaborative communities starting a joint, very likely in-person meeting. Unfortunately, that's not yet possible at the FDA campus. That will probably happen after the end of the public health emergency, but we will find a site. Site has not been determined. A possible date is provided here, but that's still up for discussion. And if there are people interested in this, you know, we will definitely have an update as a project or an event on the website. And as soon as that date has been finalized, we email this out. There's various aspects. This is probably not so much focused on digital pathology, although there are certain aspects as well, if you just think about the end stages of COVID, for example. But wanted to share this, and in case you want to share this with some of your clinical or chemistry colleagues, this would be great. Questions about this? So this is still sort of in the planning phase. If you're interested in this or working on whatever procalcitonin or some of the early sepsis markers, please feel free to reach out to Ulla, and then we include you in the planning phase. All right. The next topic, it's a packed slide, and there's a ton of links, and I will provide hopefully an adequate and neutral representation, because this is a very loaded topic. We named this in apostrophes here a valid update. For those who haven't heard about it, the FDA SLA or FDA SLA is the Food Drug Administration Safety and Landmark Advancements Act. That's part of MEDUFA. And the specific part that is included is a Title 8 Subtitle C, which is effectively the Valid Act. That's multiple 250, I think, roughly pages, starting at page 125. And that proposes to change a few things, among those adding a subchapter J to the chapter 5 of the FDAC. And as part of this, see, Shannon, 260 pages, yes. Those last 10 pages have it in it. I can vouch for that. In any case, so MEDUFA is actually proposed recommendations for reauthorization for the coming 5 years. And as you can read here, this is the formal definition of what MEDUFA is. And this is basically an act that entails another bill, which is the Valid Act. And you see I took out my prior versions, and this is the subsections of those over 200 pages from the DASLA here, and this part is basically the Valid Act of 2022. And here we will only discuss regulatory science topics, so we will not say whether it's pro or con, and we will provide a couple of links. I think if depending on which societies you follow, you will see that there were a few elements. The two things I just want to point out, again, neutral, is that collaborative communities are mentioned in the Valid Act. That has been, I think it was included since 2021, but there's also an advisory committee proposed, as well as numerous other changes. We're planning a follow-up meeting, I don't know the exact date yet, to discuss this in a more neutral fashion. And if you haven't seen the act and or some of the summaries that are provided, we will share this also. There's a couple of FDA blogs and news outlets that have reported in a relatively balanced fashion. Some of the summaries are, of course, polarizing, so take that with a grain of salt. Since this is pending legislation, we won't comment on that, but it's definitely important to look out for this because it definitely includes also software and any moderate or high risk IVCT. I know some people... Yep. Shannon, do you want to briefly comment? I saw your chat.
I'll just note, and you kind of alluded to this already, but software is included in the definition of valid as an in vitro clinical test. So if your software is being used for diagnosis, for example, it may fall into the regulations in valid versus the software as a medical device. So it is relevant to this group.
Yeah, and I think either way where you stand on for or against it, it's definitely informative to see what the FDA would think about how to, for example, conduct tech certification, and seeing that specified and refined is definitely informative, also just from a scientific perspective. I'm sure there are many comments. Ulla, did we settle for time? Not
tentatively, we're looking at June 8th at 1 p.m. Eastern.
Perfect. So maybe we can share this at the meeting update, and then if there's more need for discussion and putting out some neutral statements, that would be great. The next topic is this is from the proposed panel agenda from the American Medical Association, and in particular the CPT code subset or subsection. On the agenda for May 2022, you see that there's different dates of when these deadlines for requested materials and comments were happening, but the meeting has taken place, at least for the path and lab section, completely. And one of the things in so-called tab 44 is a set of digital pathology codes that would establish, and you see these are the preliminary X codes, as procedural codes to be reported as additional service requirements associated with digitizing glass microscope slides for primary diagnosis. So this is a proposed code set that would enable the medical encoding of slide digitization for primary diagnosis. How this will specifically interact with existing, for example, biopsy codes or 88305s for, let's say, the routine histologic processing remains to be determined. Sometimes these codes are evolving, sometimes they're not going live yet, sometimes they're being proposed just for discussion's sake, so there are many options. We do not have details, but our payer group and Esther Abels, Laura, and several people, I think I saw Eric on here, have spearheaded some of these discussions early on. And in conversations with Esther, she mentioned that it's too preliminary, but she will provide an update at the next June steering committee meeting, and then we hopefully have some additional details. I know that this is also a very controversial topic, but of course, something that's very important to accurately reflect volumes and, you know, eventually achieve some financial and or reimbursement. I don't know if someone else knows any more details at this point. But I think more to come in June. All right, truthing and validation. Brandon, can I hand this over to you?
Is there not a slide for me?
I think you provided an update. I can
I
see
know,
if I
but
find
I
this.
thought you would have that in your slide. And you want to skip me and I'll pull it up? We'll just put it in the chat.
Here you go.
There we go. That'll help. It's not open yet on my screen. There it is. Thanks for this little chance. Just so that everybody knows, this update and maybe even more expansion is at this hub space. The NCI has a collaboration space called NCI Hub. We have a group called EDAP Studies. We try and put all our updates there. I wanted to let you know that I am meeting with CPT AI community to talk about my project. And I think, you know, there are a lot of people that are interested in creating validation data sets. And so I think just like the OV Scout and using a challenge, that's all part of the same effort of the health providers and societies. That my goal is for those groups to be creating the validation data sets so that other people can then also use the same data sets so that ultimately we can compare apples to apples on algorithms. And that's been something that's always discussed here at the FDA. Somebody comes in and says, why don't we do that? And well, the law doesn't allow it. So that's why I'm trying to get actual benefactors or the people that will benefit the most from these data sets interested in creating the data sets, because they're also usually have the data set. I also want to express on behalf of my division director that we are recruiting at all levels, senior to very new. Let me know if there's anyone who is interested in DIDSER, Division of Imaging Diagnostics and Software Reliability. We have some deliverables finally making it into the published space. This first one is about a project to create training, and this will be ultimately continuing medical education credit worthy training, I believe. But there's a manuscript published in the Cancer Special Issue on TILs, and Victor presented it at Pathology Informatics. All of this material, I think, is at this link. Or at least the manuscript is at this link, and a little bit further down is a link to the Pathology Informatics presentation. A lot of that work had expert panel sessions, 8 hours, that we worked together and identified pitfalls and features and discussed how to create the training from that. But also the pathologist did data collection, and so our expert annotations are also on our GitHub. So we've shared our pilot study, which has over 7,000 annotations. Now this is our clinical expert, smaller sets of data, as well as the statistical scripts to create the figures that we're publishing so that other people could reproduce those figures. Reproducible science, that's what I believe in. The next one is a published manuscript on the analysis method I had planned for this data. Three-way mixed effect ANOVA. So it's about limits of agreement. If you know scatter plots, it's basically a way to characterize the variability in a scatter plot. But that variability accounts for reader variability. So that's the generalization. The problem is the pathologist data is not Gaussian or normal distributed. And boy, it really came home when we were looking at the data and we're pivoting a bit on our methods because we can't really satisfy the assumptions that ANOVA requires. So we're still working, and we spent 2.5 years on the methods mentioned in this paper, and now I won't be able to really use it the way they were intended. The next one is something I've mentioned already here, but I don't think I've had all the publication information. And this was a manuscript, actually an editorial about the FDA programs related to research and resources and collaboration. This group is featured pretty strongly in my work. It was a kind of a self-serving editorial as well as an FDA serving editorial, pointing to all the different tools I've been doing as part of my research here at the FDA. So please check it out. Similarly, I gave a tutorial, and I've talked about this before, on study designs and reader studies, MRMC, that stands for multi-reader, multi-case studies. This is training I gave to the FDA reviewers. I think my radiology division director, she asked me to create a video. So far I've only posted the slides online, but I think the slides are chock full of good stuff for anyone in this space. And we've talked about it a little bit here before. Then there's the presentation just given at Pathology Informatics. I kind of mentioned it, the slides are there. A poster given at AACR, kind of an overview of the project. The link is there. And then this is something I mentioned a little bit earlier, so I just wanted to make sure everyone is still pointing at and aware of the discussion in PICC on grand challenges. And then we've also got a manuscript in submission to the Journal of Medical Imaging. This one is really about the tools, the platform for data collection that we've been creating and how we've kind of been tailoring them to get better and better data. I hope I fit my time.
Perfect. Awesome. This is, showing how much is going on in this work group. And I wanted to mention a few other things. One element that may be relevance is CIDR's like putting out a newsletter, first, or issue 1 here. I think, Ola, if you could share that would be great. Awesome. Aside from some of the papers that are published from within the agency, it also shows a very detailed, including the links to the upcoming events that are organized by the various groups. So if you haven't seen this, please take a look. The next proposal here is the proposal for a cell-free working group. Again, similar to the CPIM proposal, about a recently appearing draft guidance for the early stage solid tumor detection using circulating tumor DNA. If you take a look at the table of contents, you will see that there's patient selection for molecular alterations, for residual disease, for measurement of response, and this just came out earlier this month, and it's of course a draft guidance, so up for debate, and several of the members have reached out that they would really want to review this collectively and then provide some feedback and or write probably a short position on that. So brief shout out to those who initiated this. I think we have several representatives from the vendors, and this will be a separate working group discussing this topic. Moving forward, we have the Anatomical Pathology Patient Interest Association officially joined PICC. You will see that on the website as the symbol. And in collaboration, we joined their group as well. So there should be some shared interests in all kinds of elements, and a first collaborative project was planned. The group has a strong interest in helping to streamline pre-analytic variables, and the first element is a protocol development for what would that look like when you want to systematically assess pre-analytic variables. And with their support, there may be also some sponsoring of certain projects, but the plan would be to really initiate a multi-site study And but most of it, please give a brief shout out to the leadership from APIA to joining us. So we have another patient interest association join us here. Another event happening on June 6th is a PathML presentation for an open source software toolkit for computational pathology research. This is from Renato and Jacob. Some of you know them. You see the time here, June 6th, 10 a.m. Super happy that they're presenting their tool and explaining a little bit on the ins and outs of using it. I've seen the GitHub page; it's quite intense. You will see and hopefully learn what they thought about this. And just a brief shout out that this is obviously they're very busy, but having that present to like those interested would be great. One element here as a brief update, as everyone on this call knows, you can use scientific input to change regulation. And I was invited by the European Federation of Clinical Chemistry and Lab Medicine to present and represent, as you can tell, here's a brief screenshot. This is more lab medicine, not so much digital pathology, but the concept of a collaborative community is sort of swapping over to Europe and, getting into the mode of providing scientific data to influence regulation was sort of the topic. And you see some of these people are well known in the field of AI and or lab medicine, and it was a very lively discussion. You see there's like a multi-page chat. And I presented just what we're doing with PyCC and explained a bit about the pre-competitive space and what we've accomplished. So that I just wanted to mention that's PyCC is turning Europe, it seems. At least there's a lot of interest there, which is great. I think, from a concept, any scientific input is great. As a quick overview here, before we go to the events of interest, a brief shout out. Julia Thira from the trainees group has made PyCC meeting business cards. Those are beautiful plastic cards designed to increase membership and show them to people. Look at this, if you can see it here on my screen. I think I see a heart from Kate. And, in case you're at a meeting, one of the jokes in Pittsburgh was we should always say that the second night at any digital path meeting in the hotel bar, you can identify yourself with that card. You're part of the PyCC club. In any case, so there's a few meetings coming up. I think ASCO is next. There's one in Long Island in New York. There's a meeting in Berlin, a meeting in Paris, a meeting in October, Vision. So there's a lot of meetings, and so look out for these cards. And with that, we have 3 minutes left for discussion and or other topics. And sorry that it was so packed, but it seems that people and the field is moving forward at rapid pace. Comments?
I have a question
Sure.
for Brandon regarding his approach to modeling pathology data. I don't know if he has considered Bayesian inferences. I'm just very curious to know if he has considered Bayesian approaches to looking at pathology data.
When I think of statistics, I think there's many ways to skin the cat, as they say. Kind of a horrible, I guess, expression, but whatever way it takes. Now, what I have really come to rely on is that humans are not statistical random number generators. Well, they are, but they don't fit any distributions that we have seen before. And I always fall back to ordinal data methods and ordinal statistics. And the bottom line is other people need to be convinced that what you're doing makes sense. And so the more sophisticated it gets, the more people fade, glass eye over, they don't know what you're talking about. And so I really try and use the tried and true metrics that we are very familiar with. And in diagnostics, it's sensitivity, specificity, and the ROC curve. And in quantitative medical numbers, it's the scatter plot. You know, you got to start with one of those two, or you hard to keep anybody with you. Even things like risk, you know, it's not something the guy on the street, or my colleague used to say, the person in the Washington, D.C. metro on their way to NIH. You need to be able to communicate what you're saying pretty quickly. And my hammer is the ROC method, sure. Because people are good at ranking, and especially side by side. You know, they can say this one is bigger, this one has more, but absolute scoring systems get to be pretty tough. I have my new slide for kind of my new methodology, and I'll share it here soon.
Oh, this is great.
Thanks for asking.
Thank you.
Thanks, Kingsley. If there are no other questions, 4 p.m. We made it. We will hope to see you again in June. It's hopefully not going to be as packed, and we have some time for great papers. We received a lot of submissions for like papers to be featured, but this time, unfortunately, we didn't have time to go over that because there's so much else going on. Without further ado, enjoy the rest of your day and hope to see you end of June or earlier. Bye.